Hello everyone, habari zenyu? Good morning, good afternoon, good evening. How are you doing? Welcome back. We're continuing on this series of lectures on gluconeogenesis. So we've been talking about carbohydrate metabolism, and we saw that glucose is the carbohydrates are the major sources of energy in the cells, in the human cells. And sometimes the last time we talked about glucose coming into the body from diet. There are periods when you're not eating and the blood sugar goes down. How does our body ensure that our blood sugar is usually within normal limit? So gluconeogenesis is one of the processes that allows this to happen, where you can see, just as the name says, glucose, new, genesis, synthesis. So this is actually a process of synthesis of a new molecule of glucose. Come with me as we go through this journey to understand how the body is able to make glucose. Questions we are asking ourselves: how does the body make glucose? Where does it make it from? Karibuni. So generally we've said this is biosynthesis of a new molecule of glucose, and actually some textbook will say from non-carbohydrate sources. Why from non-carbohydrate sources? Because some of the substrates that are actually used for gluconeogenesis include lactate, pyruvate, glycerol, and even amino acids, a particular group of amino acids which we refer to as glucogenic amino acids. So you can see these do not fit our classification of carbohydrates like glucose and other molecules. So that's why we say it's a synthesis of glucose from a non-carbohydrate source. The other question is, where does gluconeogenesis occur? There are three major tissues that do gluconeogenesis. One of them is the liver, which makes as much as 95%. It makes a range of 85 to 95% of the glucose. The second one is the kidney, which can make glucose during extreme starvation or in metabolic acidosis. And then we have epithelial cells of the small intestine, which can make as much as 5% of the total glucose. So only three tissues can participate in gluconeogenesis: the kidney, the liver, and the small intestine. Synthesis of a new molecule of glucose is not a cheap process. It's expensive. And we can see here, to convert 2 moles of pyruvate to 1 mole of glucose, we require 4 moles of ATP, 2 moles of GTP, and 2 moles of NADH. We can see this process is a very expensive process. So the question is, why does the body do it? So one of the reasons is that body does gluconeogenesis is during starvation, or when we have limited periods of carbohydrate intake. Gluconeogenesis is very important to maintain blood sugar concentrations. And we know there are some tissues like the red blood cells that cannot make energy, ATP, from any other source and predominantly depend on glucose. Also, tissues like the brain prefer glucose as a source of energy, although the brain can adapt to other sources in very low blood sugar levels. And then we see gluconeogenesis playing an important role during extended exercise. We mentioned in the previous class about the Cori cycle, where we saw lactate coming from skeletal muscle and red blood cells being carried to the liver, where it was used for gluconeogenesis to form a new molecule of glucose. Also, during extended exercise, we can have aerobic exercise where fat is brain breakdown. And remember, fat is stored in the form of triacyl glycerol, where to 1 molecule of glycerol, we have attached 3 molecules of fatty acid. So in the breakdown of triacyl glycerol during aerobic exercises, the free glycerol molecule can be carried in blood back to the liver and can be used for gluconeogenesis. And we're going to see this. Another time where the body needs to do gluconeogenesis is during metabolic acidosis. So during this process, there's high hydrogen ions in the bloodstream from metabolic sources. And so the kidney has to get rid of this proton and it has to pump it out, requiring energy. So gluconeogenesis allows the kidney to be able to excrete the increased number of protons against their concentration gradient. And finally, Gluconeogenesis allows the body to be able to use dietary protein in carbohydrate metabolic pathways. We're going to learn in amino acid metabolism that amino acids are not stored in the body because of the toxic amino group. So the first thing the body does is deaminate, remove the amino group and gets rid of it as urea. So what happens to the carbon skeleton? The carbon skeleton can be used for in carbohydrate pathways and even to synthesize fats, as we are going to see. And how they do this is through the process of gluconeogenesis. So let's look at how each of the substrates enter into the gluconeogenic pathway. So we saw the lactate dehydrogenase reaction, where we saw pyruvate is being converted to lactate. So in non-gluconeogenic tissues like the skeletal muscles and other cells, this reaction happens in the anaerobic glycolysis, where pyruvate is converted to lactate to ensure decreased levels of pyruvate and the oxidized form, and to ensure adequate levels of the oxidized forms of NAD, which will allow glycolysis to continue. However, in glucogenic tissues like the liver, the kidney, and the small intestine, the reaction will be happening in the reverse direction. We have an isoenzyme of lactate dehydrogenase, which will convert lactate back to pyruvate. However, also in gluconeogenic tissues, the direction of the reaction will depend on the ratio of the oxidized NAD and the reduced NAD. However, when lactate is carried to the liver, lactate will be converted back to pyruvate. I think we saw this when we looked at the Cori cycle. In the Cori cycle, we saw skeletal muscle breaks down glucose either from bloodstream or from stored glycogen and breaks it down to pyruvate. And in cases of strong contractility, where there is low blood supply, it is converted to lactic acid. This lactic acid is carried in the blood to the liver, where we've seen the lactate dehydrogenase will convert it back to pyruvate, and pyruvate will be used to synthesize glucose through gluconeogenesis. And the glucose can be carried back into blood to skeletal muscle, where it can continue providing energy for contractility. So we mentioned that Cori cycle operates between the muscle and the red blood cell and the liver. Another metabolite or another substrate for gluconeogenesis is alanine. So alanine is a representative of the amino acids. So where does alanine come from? So in glycolysis, we can have the end product being pyruvate, for example, in muscle. And one of the things that happens to this pyruvate is that pyruvate can be aminated, or we can add an amino group to pyruvate through transamination reaction. And when we add an amino group at the carboxyl group of pyruvate, we form the amino acid alanine. Now, this alanine that is formed in muscle from pyruvate is released into the bloodstream and is taken back to the liver. And in the liver, the alanine can be transaminated again, can undergo reverse transamination back to pyruvate. And then pyruvate can be used for gluconeogenesis. So we can see a common intermediate from alanine and lactate is pyruvate, which will be used in gluconeogenesis. Another intermediate is glycerol. Where is glycerol coming from? We have agreed that fats are stored in adipose tissue in the form of triacyl glycerol, where 3 fatty acids are attached to a molecule of glycerol to form the triacyl glycerol. And during catabolism or during starvation, where blood sugar is low, fatty acids, triacyl glycerol is used to provide energy. And one of the things that happen is that the fatty acids are detached from the glycerol. And the free glycerol is released into blood. And once in blood, is taken up in the liver. And the liver can phosphorylate the glycerol to form dihydroxyacetone phosphate. Or dihydroxyacetone phosphate can be used for the synthesis of glucose through the process of gluconeogenesis. Okay, so of course, in the presence of low blood sugar, the liver will be making glucose, and so gluconeogenesis will occur. The other intermediate we talked about are amino acids. So we know all amino acids have the amino group, which is attached to a carbon group. So who are gluconeogenic amino acids? Gluconeogenic amino acids, we classify them as amino acids which, once we deaminate them, we remove the amino group. The carbon skeleton that remains is either pyruvate or an intermediate of the TCA cycle. So in this particular diagram, you can see alanine, cysteine, all these. When you deaminate them and if you break them down, one of the intermediates they will give us is pyruvate. We have some amino acids, once deaminated, can be giving us alpha-ketoglutarate. Others can give us succinyl-CoA, for example, isoleucine, threonine, and valine. Others can give us fumarate, and others can give us oxaloaspartate, for example, aspartate. So long as they become one of these products, they can form oxaloacetate, and we're going to see how oxaloacetate can be used in gluconeogenesis. So we say glucogenic amino acids are the ones, when they're deaminated, give us either pyruvate or one of the intermediates of the TCA cycle. Okay, and so long as they form this, through oxaloacetate can be used through the process of gluconeogenesis. The other amino acids are ketogenic amino acids. Ketogenic amino acids give us either acetyl-CoA or acetoacetyl-CoA. Okay, and they don't give us a product of the TCA cycle. These are referred to for ketogenic amino acids because acetyl-CoA during starvation can be used to form ketone bodies. We're going to see this when you talk about amino acid metabolism. A quick challenge, you can see some amino acids are both ketogenic and glucogenic. For example, look at isoleucine. So that means when they are broken down, they'll give us both a ketogenic product and a glucogenic product. Okay, I just take a moment and look which other amino acid is both glucogenic and ketogenic, and which other amino acid is either exclusively ketogenic or exclusively glucogenic. Just pause and look at this picture a while and see which amino acids are glucogenic, ketogenic, exclusively ketogenic, and both glucogenic and ketogenic. Okay, so which are these reactions of gluconeogenesis? Now that we've seen how the substrates end up to pyruvate, how does gluconeogenesis occur? So this is very interesting. Gluconeogenesis occurs in the cytoplasm, which is the same compartment that glycolysis occurs. If you remember what we talked about glycolysis, in glycolysis there were 3 irreversible reactions and there were 7 reversible reactions. Reversible means we can go back and forth and we move to one direction depending on the concentration. Okay, so the 7 reversible reactions of glycolysis are also used in gluconeogenesis. Okay, so both gluconeogenesis and glycolysis share the 7 reversible reactions. However, in glycolysis, we had 3 irreversible reactions. And in gluconeogenesis, these irreversible reactions are overcome or circumvented by 4 alternate reactions. So if we come down here, if we look at glycolysis, the end product was pyruvate, okay? And this reaction of pyruvate kinase was irreversible. But all these reactions of enolase, phosphoglycerate mutase, phosphoglycerate kinase, glyceraldehyde 3-phosphate, triose phosphate isomerase, and aldolase were reversible. Then we have the PFK reaction that was irreversible, and then we had the phosphoisomerase reaction which was reversible, and the hexokinase step which was irreversible. So this irreversible step, the hexokinase, PFK1, and pyruvate kinase reaction are overcome by 4 steps. However, these reversible steps are shared by both glycolysis and gluconeogenesis. That's why both of them occur. Most of the parts occur in the cytoplasm of a cell. However, in gluconeogenesis, we're going to see some of the reactions that occur in the mitochondria. So let's look at these 4 reactions of gluconeogenesis that differ from those of glycolysis. So one of the reactions that differ is how do we convert pyruvate to phosphoenol pyruvate. Remember, this reaction was a high energy reaction where the energy for transferring the phosphate group from phosphoenol pyruvate, the energy that we got, we created enough energy to form ATP. Remember, pyruvate plus ADP gave us phosphoenol pyruvate plus ATP gave us pyruvate and ATP. Okay, so the phosphate group was moved from phosphoenol pyruvate to ADP to form ATP. This was a substrate level phosphorylation. So there was a lot of energy that was released. It was an hexagonic reaction. So to overcome an hexagonic reaction, the gluconeogenesis has two steps. The first step is catalyzed by an enzyme called pyruvate carboxylase. Pyruvate carboxylase carboxylates pyruvate to end up forming oxaloacetate. Okay, and it requires energy to form this reaction. Energy is required to create this new carbon-carbon bond. Okay, and this is catalyzed by an enzyme called pyruvate carboxylase. Pyruvate carboxylase enzyme requires biotin as a coenzyme, as a cofactor, as a coenzyme, as a prosthetic group. Okay. In addition, it requires the cofactors magnesium and manganese. So you can see the pyruvate carboxylase does the first reaction of converting our 3-carbon pyruvate to a 4-carbon oxaloacetate by carboxylation reaction. Of interest is that this reaction occurs in the mitochondrial matrix. This carboxylation occurs in the mitochondrial matrix. So what that means, once pyruvate is carboxylated to oxaloacetate, oxaloacetate has to move into the cytoplasm where the rest of the reaction of gluconeogenesis will occur. So how does oxaloacetate move? Oxaloacetate, we know it cannot traverse the mitochondrial membrane. We said it is virtually impermeable. So for it to be carried back into the cytoplasm, it has to be carried in the form of malate. If you look here, what happens is that we have oxaloacetate in the mitochondrial matrix. Okay? Now, this oxaloacetate in the mitochondrial matrix is what we want to carry into the cytoplasm. How does this occur? So, malate will be transaminated with alpha-ketoglutarate, and when we add amino group to oxaloacetate, we form aspartate. Aspartate can move back into the cytoplasm, and the reverse transamination can occur to give us oxaloacetate. Now, this oxaloacetate can be used for gluconeogenesis in the cells. In the other hand, we can get oxaloacetate from the cytoplasm being converted to malate, and the malate can be carried back to the mitochondria to be used in the electron transport chain. So how oxaloacetate moves from the mitochondria to the cytosol, or from the cytosol to the mitochondria, is through the malate aspartate shuttle. For example, if we want to use the malate here for gluconeogenesis, we can have the malate, the oxaloacetate being converted to aspartate to come into the cytoplasm, where it is used for gluconeogenesis. So an important enzyme here, you can see the malate alpha-glutarate carrier and the aspartate glutamate carrier. So we have that shuttling of between the mitochondria and the cytosol of oxaloacetate in the form of malate. So this enables not only oxaloacetate to move across, but also allows the reducing equivalents that we get, for example, from glycolysis to be carried into the mitochondria to be taken to the electron transport chain. So the malate shuttle transfers not only oxaloacetate, but also transfers reducing equivalents from NAD H, sorry. So once the oxaloacetate gets into the cytoplasm, now remember it was formed in the the mitochondrial matrix. Once it gets into the cytoplasm, oxaloacetate now can be converted to phosphoenol pyruvate. Okay. However, this conversion involves a decarboxylation of oxaloacetate and addition of a phosphate group. This phosphate group is derived from GTP. You can see here, GTP donates its phosphate group to give us phosphoenol pyruvate, and in the process we lose carbon dioxide. This reaction is catalyzed by an enzyme called phosphoenol pyruvate carboxykinase, PepsiK. Okay, and it requires manganese for its activation. And it's very interesting. Now you can just see in the two steps, from when you converted pyruvate to oxaloacetate, then to phosphoenol pyruvate, they have occurred in two different compartments. The pyruvate carboxylase reaction has occurred in the mitochondria, and this phosphoenol pyruvate carboxykinase reaction occurs in the cytoplasm. And in the pyruvate carboxylase reaction, we required ATP, which provided energy. And in this PEP carboxykinase reaction, we required GTP, which was the donor of the phosphate group, to form phosphoenol pyruvate. So these two reactions, the pyruvate carboxylase reaction and this phosphoenol pyruvate carboxykinase reaction, are together combined, used to convert pyruvate to phosphoenol pyruvate, and their purpose is to overcome the 10th irreversible step of glycolysis catalyzed by the pyruvate kinase reaction. So from here, the reactions will go together reversibly, back, until we reach the point of fructose 1,6-bisphosphatase. So that's why we are saying this conversion from phosphoenol pyruvate to fructose 1,6-bisphosphatase will go through the reversible reactions. However, remember, we'll require two pyruvates to be converted so that by the time they reach the aldose reaction, we have the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate, which can be combined together to give us our fructose 1,6-bisphosphate. These were reversible reactions of glycolysis. Now, to form fructose 1,6-bisphosphate from fructose 6-phosphate, we have the phosphofructokinase reaction 1, which we said was the main rate-limiting step of glycolysis. So this reaction, like the pyruvate kinase, was an hexagonic reaction. It released a lot of energy, so it is not easily reversible. To overcome this step now, gluconeogenesis has a new enzyme called fructose 1,6-bisphosphatase. So in this particular reaction, the fructose 1,6-bisphosphatase uses a molecule of water and splits the phosphate from carbon number 1, and we go back to get our fructose 6-phosphate. Okay, so it does a phosphorylytic reaction, or it's a hydrolytic reaction which uses water. So fructose 1,6-phosphatase is an allosteric enzyme, and is the major regulatory steps of gluconeogenesis. Okay, and we saw citrate inhibited PFK1, but we can see it activates fructose 1,6-bisphosphatase. And the inhibitors of PFK1, AMP and fructose 6-bisphosphate. In the other hand, the activators of PFK1 are inhibitors of fructose 1,6-bisphosphate. So this is very important because even though both glycolysis and gluconeogenesis occur in the cytoplasm, they cannot occur at the same time. So you can see modulators that activate gluconeogenesis at the same time will inhibit glycolysis. So the next reaction, PFK to glucose 6-phosphate, was catalyzed by phosphoglucoisomerase, and this was a reversible reaction. And the next reaction was conversion of glucose 6-phosphate back to glucose. So this reaction is catalyzed by an enzyme called glucose 6-phosphatase, very similar in action as fructose 1,6-bisphosphatase, where it adds a molecule of water and this does a phosphorylasis by splitting the phosphate group and giving back our free glucose. So this particular enzyme, glucose 6-phosphatase, is only present in gluconeogenic tissues. It is not present in muscle and all that, okay? So it's only present in gluconeogenic tissues, and we're going to see it again when you talk about glycogen metabolism next. So remember, only free glucose can move back into the blood through the GLUT2 receptors present in the liver. Okay, so this reaction is what allows free glucose now to go back from the liver into the blood stream during periods of starvation. So this is our summary. We have our pyruvate, 2 of them. They are carboxylated in the mitochondria by pyruvate carboxylase, requiring energy to give us oxaloacetate. Then oxaloacetate gets decarboxylated and phosphorylated by PEP carboxykinase. This reaction will be occurring in the cytoplasm, and again we require GTP. Then phosphoenol pyruvate up to fructose 1,6-bisphosphatase reaction, we have reversal of glycolysis. Okay, and then we have fructose 1,6-bisphosphatase, which dephosphorylates the fructose 1,6-bisphosphate to give us fructose 6-phosphate. The reverse isomerization to give us glucose 6-phosphate, and to overcome the exokinase reaction, we have the glucose 6-phosphatase, which dephosphorylates glucose 6-phosphate to give us glucose. So we can see the 3 irreversible steps of glycolysis are overcome by the 4 reactions. And we can see when in glycolysis we release energy, here we add the energy. So how many? To form 1 6-carbon molecule of glucose from 2 3-carbon molecules of pyruvate. How much ATP required? We have these 2 plus 2, those are 4 ATPs. How much GTP? 2 GTPs, and we needed to add a molecule of NADH. Okay, so it's actually a very expensive process to synthesize 1 molecule of glucose from 2 molecules of pyruvate. So how does gluconeogenesis be regulated? We have mentioned slightly about it when you talk about the reverse regulation, but let's look at it in a bit more detail. We can have the non-hormonal regulation, which occurs minute by minute. One of the ways depends on the substrates. So if we have high concentration of the substrates, for example, in the liver, pyruvate, lactate, alanine are all coming from muscle or other tissues to the liver, and in high concentration, the liver is going to use it for gluconeogenesis. Okay, so high concentration of substrates will allow gluconeogenesis. Another is allosteric regulation, where we had modulators like citrates. Okay, so acetyl-CoA is a concentration-dependent regulator of the pyruvate carboxylase. We know pyruvate carboxylase is the first enzyme for gluconeogenesis, and it is located in the mitochondria. So acetyl-CoA will stimulate action of pyruvate carboxylase, but we have seen other modulators like citrate, AMP. Then we have the hormonal regulation. We've said gluconeogenesis will occur during periods when we have low blood sugar. So the hormones glucagon, epinephrine, and the glucocorticoids like cortisol, the main purpose is to increase the blood sugar. And one of the ways they're going to do this is going to stimulate gluconeogenesis by stimulating increased synthesis of the enzymes of gluconeogenesis. So once we have high blood sugar, we have low blood sugar, glucagon, epinephrine, and glucocorticoids will stimulate high of the enzymes for gluconeogenesis, especially these 4 enzymes that overcome the 3 irreversible steps of glycolysis. Another thing is that insulin suppresses the synthesis of this enzyme. So if you have a high carbohydrate diet and the blood sugar is high, okay, the insulin will inhibit gluconeogenesis. However, this is not the case in diabetes. I challenge you to think about it, why we still have gluconeogenesis even in diabetes. Another way we talked about, but let's look at it a bit more, we mentioned about reciprocal gluconeogenesis. such that even though both gluconeogenesis and glycolysis occur in the cytoplasm and share the enzymes, they do not occur at the same time. Okay, so we saw the major point of reciprocal regulation was at the PFK1 and the fructose 1,6-bis phosphatase reaction. So the PFK1 or the phosphofructokinase reaction 1 converts fructose to fructose 1,6-bis phosphate, and it gets the phosphate group from an ATP. We saw when the fructose concentration of fructose 6-phosphate is very high, another enzyme, phosphofructokinase 2, is activated and it phosphorylates fructose 6-phosphate to form fructose 2,6-bis phosphate, which is a positive modulator of PFK. However, this fructose 2,6-bis phosphate, while it activates PFK1, it inhibits fructose 1,6-bis phosphatase, such that when PFK1 is active, fructose 1,6-bis phosphatase is inactive. However, ATP citrate will inhibit PFK1 but will activate fructose 1,6-bis phosphate, signaling to the cell to start synthesizing glucose through gluconeogenesis. In the liver, ATP and alanine will inhibit pyruvate kinase, okay, while ADP inhibits pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So we can see this is what we mean about reciprocal regulation and plays a big role to make sure gluconeogenesis is not occurring at the same time as glycolysis. We have some diseases that can be associated with gluconeogenesis. One of the diseases is called multiple carboxylase deficiency. We've seen pyruvate carboxylase, the first enzyme of gluconeogenesis. The carboxylase enzyme all depend on biotin. So you can see this enzyme, pyruvate carboxylase, acetyl-CoA carboxylase, propenyl-CoA carboxylase, and beta-methyl-crotonyl-CoA carboxylase all require biotin. So how is biotin added to this enzyme? It is catalyzed by an enzyme called holocarboxylase synthetase enzyme. So this is the enzyme that adds biotin to this carboxylase enzyme to allow them to work. However, in this deficiency, there is lack of this enzyme, and this makes it impossible to add biotin. And when biotin is not added, these biotin dependent enzymes are inactive. And so this presents with retardation, developmental retardation, ketoacidosis, hair loss, and erythematous skin rash. Luckily for us, this condition can be managed by adding therapeutic doses of biotin. Remember, this is not absolute absence, it is deficiency. So maybe the copies are not require high concentration of biotin to be able to add them to these enzymes to allow them to be active. Another disease is von Gieck's disease. We are going to talk about this as one of the glycogen storage diseases. It's also known as glycogen storage disease type 1. Here, there's deficiency or defect in glucose 6-phosphatase, meaning now glucose 6-phosphate cannot be converted back to glucose. If you think about this occurs in the liver where the liver is the major site of gluconeogenesis. So if the liver does the gluconeogenesis and reaches glucose 6-phosphate and you cannot convert it back to glucose, this glucose 6-phosphate will be used for other pathways, like for example glycogen synthesis or the hexose monophosphate shunt, whose one purpose is to form ribose, which can be used for nucleic acid synthesis. So you'll find that the liver can enlarge because of accumulation of a large amount of glycogen. Okay, the blood sugar will be low because gluconeogenesis, remember, its purpose was to increase the blood sugar, especially in periods of not eating. So the person will present with hypoglycemia because the liver cannot release glucose to relieve the symptoms of low blood sugar. Another thing is that there will be hyperlipidemia because apart from glycogen synthesis, the liver will have another pathway, HMP shunt, in an attempt to reduce the glucose 6-phosphate concentration. One of the products of HMP shunt is large amounts of NADPH, which are used for anabolic synthesis, and so we'll have increased synthesis of lipids, okay? But also because of low blood sugar, because the cells are not synthesizing the blood sugar, the body will start breaking down lipids, and this is what will cause hyperlipidemia. And then one of the products of the pentose phosphate pathway is ribose, and ribose is used for the synthesis of nucleic acid, but this for the synthesis of nucleotides. But these nucleotides are not needed by the cells, and so they are broken down, and especially purine nucleotides will give us uric acid and will cause hyperuricemia. We're going to talk about von Gehr's disease in much more details as we discuss glycogen storage diseases. So as we end, I would like us to look at this case study where I would like you to spend some time thinking about it. A 2-year-old boy is admitted to hospital with severe lactic acidosis. He has a history of poor growth and development, repeated seizures, ataxia, and persistent metabolic acidosis. Urinalysis indicates high levels of organic acids, pyruvate, lactate, and beta-hydroxybutyrate. Luckily, the patient is administered with 10 mg of an oral dose of biotin per day. The metabolite levels return to normal within 3 days, and the more general symptoms such as ataxia improve over a period of several days following therapy. So think about it. Why is biotin important for metabolic pathways like gluconeogenesis? And how might a deficiency of biotin disrupt the energy production in the body? How is this deficiency leads to buildup of metabolites such as lactate and pyruvate? So this is your revision question. Think about it. How does biotin, which we saw was important for pyruvate carboxylase, cause increased amounts of lactate and pyruvate in blood? Otherwise, this is the end of gluconeogenesis. Thank you for your time. See you in the next one.